Tara, thank you. Tara asked me to give an overview of what we did and then comment a little bit on Science Today and what it means for your students. So I start off with, who am I? Tara, I guess, took care of a lot of that. I am the Center Director for Epigenetics here, but first and foremost, I'm a scientist. I'm a professor. Like you, I'm a mentor. I teach every day in one sense or another. I lead the institute. I try to shape policy and research strategy for the future and see into the future to shape, one, the research directions we choose and how we shape, for instance, the graduate school. So here we have a completely independent, nationally accredited graduate school that hands out its own PhDs, and it makes us a very autonomous, independent place as far as our research goes. How did I get to science? It's not glorious. The truth is I didn't get into medicine, right? My parents pushed me in that direction. I always loved biology. I always loved medicine. I found my grandpa's surgical tools one day in my grandparents are Czech, so I was in Czech Republic, and I opened this bag of surgical tools with a giant syringe, and as a kid, I'm like, this is great, right? Truth is, I got lucky. So I think seeing patients every single day is tough. I think it's really hard. Getting to come to work here whenever I want, I can show up at 8 or I can show up at 10. That's a different ballgame than having to run a practice with patients coughing on you every single day. So a little bit of a blessing in disguise that the medical school system here in North America is actually so hard to get into. So that's the honest version of how I got here. And I think the most important for me, what I love to talk about is what do we study? And we study this kind of idea. These are dandelions, right? That's the right English name. They're pretty identical, right? They grew up in different fields, maybe. Well, these two probably right next to each other. But no matter what field you take them on, they look exactly the same, right? It's a pretty powerful concept. And I'm a disease biologist, or as Tara said, a complex trait biologist. So any of our traits, like the color of our hair, the color of our eyes, the weight of our arm, the strength of our muscles, they're regulated by tens, if not hundreds, if not thousands of genes, right? But somehow all those processes end up in like a picture-perfect replication of the same genome, right? And so this is, I think, a perfect visual example of that. This is the cover of a photography book by Martin Schuller. He's a very famous portrait photographer. He's German, he's got a studio in New York, and it's called Identical, and it's portraits of identical twins. And so you as teachers probably have had much more interaction with twins than I do, because I have each of my students I have for 5 years, and I only have a few students. You guys get the joy of having, hopefully not 30 people in your classroom, maybe, but I guess you're always trying to hold it down. The wonder of this book is how beautifully he's taken these images. And as a scientist who likes studying identity and differences, the lighting is always perfect. He's always standing the same distance away from the image. And these are two sets of quadruplets he's photographed, this set of triplets. And if you look, it's amazing. These people start out as one cell, right? One egg and a sperm come together and they form the zygote, this one single cell. And that turns into trillions of cells after 30 years, and it all comes out the same, right? After every different environment, some are playing tennis, some are eating hamburgers, right? And it all comes out relatively similar. But if you look carefully, you can see differences, right? So this is the two girls that are on the cover of the book. And I think it's pretty tough to find differences. Even like the areas where her freckles are kind of at their densest are the same on both girls. So, sort of how did the skin cells over here know to be similar to the skin cells over here on a totally different body, right? You can see some differences. And I'll show you actually 3 twin pairs. I don't know, can you see the differences? I can see some differences, but I'm a bit of a weirdo, a pattern person. She's got a broader facial structure. It's very hard for me to see her. Broader facial structure, right? Slightly different eyes and thinner lips, right? And I'm going to show you 2 more twin sets. Same thing, hey? the one on the right has a slightly broader face, slightly different eye. The eye shape's a bit hard to see. Her lips aren't so thin, but these two. It's not the haircut, don't be fooled. It's broader facial structure, slightly thinner lips, and slightly different shape of the eyes. So, if twins can be different, and they can be, I showed you, I've easily found 3 pairs that have the same differences. That kind of implies that you and I could have been different, right? Because each of us could have been a twin, right? So we could have come out as us, and this kind of implies we could have come out as a different person. Wouldn't you love to know who that other person is? I do. I want to know who Andrew 2 is. What would our personality have been? Would we have behaved the same? Would we have behaved different? How many people here have had identical twins in their classroom? Well, you see, you've had much more interaction with identical twins than I have. How many people have had identical twins that were easy to tell apart? Yeah, quite a few, right? Twins are not exactly the same, right? They do have differences. And that was about, from my perspective, that was about half of the hands, right? So most identical twins are, but really a substantial portion are quite different. And this boils down to our favorite topic, which is epigenetics. So, the concepts that are around genetics that do genetic-like things, but that aren't coded in your DNA sequence. That's basically the definition. And they have, actually, in nature, we have examples where they really have profound effects on the body. And this is one of the most classic example. Does anybody know what that is? Queen bee and a worker bee, right? They have the exact same DNA. Actually, the worker bees are basically clones haploid clones of the queen. But you can see almost everything you look at, and when you compare the queen and the worker bee, almost every single thing is different. The hairs on her back, right? The color of the abdomen, the length of the abdomen, the shape of the wings, the size of the wings, everything. The behavior. One stays in the hive, hides, mates, lays eggs, eats, does nothing else but stay there. The other never mates, goes out of the hive, works all day. Nothing is the same about these individuals except for the fact that they have 6 legs, a head, 2 wings, etc., that kind of thing. And this kind of striking difference on the exact same DNA template is visible no matter where you look in nature. You can find these things. So it's not really unique to insects, though almost all insects show it really well. So, we all know about the locust, right? There's the kind of swarming form that has biblical connotations to it. And then there's this solitary form that when there's not many of them around, they just sit there and eat, and they're gentle creatures. There's multiple types of fish where you can get essentially the same DNA blueprint, and you get vastly different colors and patterning. This is Daphnia. It's a model organism that has sort of a normal and then a defensive shelled version that has a little spike on it. The famous horned beetle. I'll show you a bigger picture of these two in a sense. There's a worm. This worm here, it's a parasitic worm. The eggs get laid onto beetles, and they sit there in a kind of dormant state on the beetle until the beetle dies. And then this is what takes care of eating the dead beetle carcass. And what's really interesting about this worm is depending on the density of eggs that are laid on the body of the beetle, the worms either come out in this version, that as you can see has a tooth, or this one. So if there's not many eggs, they come out this way, and they're herbivores. They basically eat bacteria and fungi that are eating the beetle. If there's lots of competition, they come out this way with a tooth, and they start eating each other, right? And they start competing with other worms. And you don't get a worm that has half a tooth, right? You don't get anything in the middle. The DNA blueprint has, at some point, there's a developmental switch, and it either goes this way or that way, just like the queen bee, worker bee kind of idea. So these ideas are not rare in nature. And if you remember those twins, I said 3 of those twin pairs kind of had reproducible differences. They had the same differences between them. So it's intriguing to think, could we have been version A and version B? Okay, so this is kind of how we conceptualize this idea. In this case, I've used humans, and I've made some larger than the other because I study metabolic diseases, type 2 diabetes and obesity. Let's forget about the Weissenberry. It's not important. The key thing is this idea of non-Mendelian variation. So the variation that we talk about today, or these switches, etc., these are things that don't follow Mendel's laws. They don't follow the laws of genetics. So if I take the same DNA blueprint and one of the individuals gets heavier, and then they pass their genes on to the next generation, everything gets reset. With Mendel's law, that doesn't happen. If you have a mutation, you don't reset the mutation. The mutation is passed and maintained. If that DNA molecule was given to the offspring, that mutation or everything coded in that DNA molecule stays, right? But as I told you, there's two different, right? There can be multiple body plans in a piece of DNA. So these are both male dung beetles. Can anyone guess the one thing that drives the difference between these two? Yes, that's right, the size of the poo ball that they were born into. So dung beetles, the eggs are laid into a poo ball, typically a rhinoceros poo ball, and they grow and develop. And if it's a small poo ball, this is the kind of beetle you get. If there's a large one, this is the kind of beetle you get. Like if there's lots of food, you get a big-horned beetle, male. These are two males. But there are medium-sized poo balls out there, right? And that's the point I wanted to make before, right? In the medium-sized poo ball where an egg is laid, the organism either gets a horn or it doesn't. It doesn't get a medium-sized horn, right? So there's some switch. Right, so how about if this could happen with humans, these developmental switches? And we have some precedent data that these kind of things can occur. You know, who would we be? That's an interesting question. But how about disease susceptibility, right? So for the half of the room that has twins that they could easily see the difference between. I mean, I'm talking about identical twins, about monozygotic twins, right? Maybe one's going to get a disease that the other one's not. So how does that happen? That's what really interests us. And I think we're still in science at this point where we question, can this really happen? I think twins are the greatest example that it can. We have some more that are sort of more disturbing. It's the only disturbing image I'll show, and it's from World War II. In the Netherlands, at the end of World War II, there was a famine where calorie intake for every person was reduced to about 800 calories per person, and this included pregnant women, right? So can you imagine for those of you who are mothers, being pregnant and only getting about a third of the food you need? No Haagen-Dazs ice cream on the couch in the evening. So the most interesting thing about this unfortunate natural experiment is the children that were so the fetuses that were inside the womb during that time, 50 years later, so nowadays, they're 60, 70 years old, they have a much higher incidence of diabetes, obesity, and heart disease than those children that were born just 3 months before or after they were, right? So there was something about being in the womb at a period of starvation that changed the course of their life. Nothing was noticed, for most of their lives, but then come middle age and approaching seniority, rates of a lot of Western diseases went up, right? And now that we have epidemics of Western diseases, we wonder, well, how much of this is genetic and how much is actually just programmed, whether it's in utero, whether it's during pregnancy, or whether it's other environmental factors. So how does it work? So this is all the result of, as I said, what we call epigenetics. Other word we use for it is developmental programming, because it's a child that's in the middle of very sensitive time points of development, that somehow the body is being reprogrammed for lifelong control. The other word that we use to describe it is canalization. And epigenetics, sorry, there's an S missing, mediates canalization. So what is canalization? Well, you can kind of think of it, right? We channel the potential of a DNA blueprint, right? So like I said, twins, they have the same DNA blueprint, and we'll get the same product. You can think of it kind of as a machine that eats DNA and pumps out little identical clones. Because in a sense, that's a bit of a joke, but researchers have cloned animals, right? Dolly the sheep was the first animal that was cloned, and it made a lot of proof of principles for scientists and for the power of genetics, right? Inbreeding, right? In mountain valleys of Italy, there's certain families that have are almost genetically identical, right? It's a geneticist's treasure chest, right? Because then certain mutations get locked in. They're very easy to map. You can map which disease came from exactly which gene in the genome. But what cloning, what those Mountain Valley natural experiments show us is that power of DNA, right? Just like the twins. So people have been studying the plasticity, so the sort of dynamics that a single DNA template can have for almost 200 years now. One of the most famous early examples, who's been to Vienna? 1, 2, 3. There's a giant park in Vienna known as the Prater, a beautiful place. It's 10 square kilometers. I don't know what that is in square miles. 3 square miles, probably 4 square miles. And it's a lovely park. At one end is the Ferris wheel and the amusement park. This Ferris wheel still exists today. This building is not there. This building was the vivarium. So, a place where life is studied. And scientists there at the time, and this is late 1800s, were already building many contraptions. This is from a book about the vivarium, to control environment, that they could take insects, animals, plants, and grow them up in identical conditions. They could control it as much as they could and look at the output. And even then, so over 100, now it's 150 years ago, the researchers had shown that if you adjust the environment of developing butterflies, you could change the wing patterns. You could change the size and the wing patterns, right? And this was true across almost every single species they test. If they raised the temperature a bit, they could make the wings better. If they made the temperature down a bit, they could change the coloring of the wings, right? And these are the 3 most prominent scientists who were there. And so they had the audacity to conclude in their book that for all major groups of plants and animals that exist, that they had tested, environmental changes in parents. So they even took just the parents, not even the developing organisms, but the parents, that environmental changes in those parents could produce changes in the offspring, even when those offspring grow up in normal conditions. Right? And so that the Dutch hunger winter idea is a little bit the same, although there were also the fetus was being starved. But we even know that if you have exposures, whether environmental, dietary, etc., to parents before a child is conceived, you can pass on information. Right? And that's a bit crazy. And so they asked 150 years ago, now the question is open, by which mechanisms the modification of the offspring is induced. And here we are today still trying to figure out the same question. So that tells you how slow science is. 150 years, same problem. We are closer. But one of the reasons not hardly anything, that's unfair. One of the reasons we don't know is because along came genetics. So at that time, we didn't know what a gene was. We didn't know what DNA really was. We didn't know its structure. Hadn't been discovered that there was this alpha helix, that there was base pairs. The base pairs coded were like letters of an alphabet, right? And the problem with genetics is those identical twins, right? They're so reproducible that the genetics world kind of exploded and said, well, everything is in the genes. All variation between us is defined by your genes. And I've shown you now multiple examples that that's not true. Queen bee, worker bee, horned beetle, not horned beetle. So nowadays we conceptualize the differences between each one of us from coming from 3 categories. One is really true genetics. So now we're slowly beating down the genetic cysts. And it is responsible for about 50% of variation, but the rest comes from 2 other parts. One that we call epigenetics. We also call that chromatin, because that's the structure in which DNA is held, and I'll show you a little bit about why we think that's important. And the other is environment. And of course, some of our phenotype, some of our output is in the environment, right? You eat a lot of cheeseburgers, eventually they catch up with you, right? Okay. So these kind of epigenetic differences that arise can happen at a whole organism level, and it arises from differences that happen in cells, right? The same process, where is your DNA? 1 molecule of DNA, or 2 molecules of DNA per cell, right? The cells are where all the action is. So upstairs here in the Department of Epigenetics, you'll find that some people study cells, some people study organisms, and some people just study the sort of naked chromatin. Okay, how does it work? This is the same thing I've just said. We have our DNA. I think we'll all remember that, right? The alpha, the double helix. DNA is wrapped around sets of proteins called nucleosomes. And this is what it really looks like down an electron microscope. It really is what the first discoverers of this phenomenon called it beads on a string. It really looks like beads on a string. So the little ball is a ball of very specific proteins. DNA is wrapped around it twice. And then this structure here is then kind of twisted and wraps around itself and forms that structure there. Right? And this is kind of the limits of resolution that we can see, so I can't get you a clearer image. The rest you have to leave up to artists, and then suddenly everything looks very pretty. Right? That's then coiled up further, and you eventually get a chromosome, right? The whole single piece of DNA. And just to remind you, in one cell, so this is the nucleus of a single cell that's about 10 microns apart, there's 2 meters of DNA stuck, right? And that process that I showed you, the beads on the string and wrapping it and coiling it very specifically without making any knots, is how it's all possible. To unwind it, express a gene from the DNA, and then package it up again without creating a knot. It's phenomenal. I'll never be able to kind of fathom how that works 1 trillion times in each of us, in each cell, at 25,000 different genes within, So this is a cell nucleus. The lighter stuff is loosely packed DNA. The dark stuff is the super coiled and then coiled DNA. And this is then what an artist would say it looks like, right? The coils. And we study those the interactions between the kind of dense stuff, the light stuff. How would you make a difference between twins? Well, if you make some genes a little bit more compact, they get harder to be expressed. You get a little bit less protein from that gene, and suddenly you have a difference throughout the whole organism of one protein. That's right, the functional unit. Okay. At chromatin, this is another diagram of it, there's about 1,200 proteins. So we have kind of 1,200 opportunities on how, and all their interactions, how to control that structure, right? And that then, I won't show you more pictures, that, you can imagine what this side of the picture looks like, has sort of hundreds of inputs from metabolism, for instance, how we can change that structure. Okay, I'm going to give you just 1 or 2 examples from our own work, and that's one where we kind of lose the middle ground and we get something like the beetle example, so the case of bistability. So just that what you take away from this is these kind of switches are not the only thing epigenetics does. So if this is kind of our most probable genetic output, epigenetics can push it 1% off kilter, 5%, 10%, 90%, or it can flip, kind of flip switches. Right? So both are possible. Both are made by epigenetics. And I think this has been the struggle of the field, is even in science, we like things boiled down to super simple, pathway diagrams that we can easily talk about. And so this is a problem, is epigenetics does 2 things, totally different, and sometimes it does a mixture of both. A few more precedents for this case, because we think this is happening in humans, and it would have huge disease consequences. Until very recently, it was not believed that this could happen in mammals at all, in any mammals. There is a precedent, and that's the naked mole rat. So these are 2 naked mole rats that are highly inbred, genetically identical. So it's another interesting example. So if you take a colony of naked mole rats, they all live underground, there's a queen. The queen gives off many, many offspring, genetically essentially identical. And they all come out like this. This is also kind of the worker type. When in times of plenty, when there's lots of food, then every 30th individual or so will look like this, right? And so this is a CAT scan, and you can see. Look at the cheeks on this. These are huge fat pads here. So they got really chubby-cheeked, but much bigger. You can see the bones are even stronger built. You get a bigger, what they call morph, and it's a dispersal morph. And so I challenge you, why would nature make a second type of naked mole rat? Only in times of plenty. What is possibly the reason for this? So what's the problem with having a colony? Is you inbreed, right? the problem with inbreeding is if you have a mutation, you lock it in and the whole colony gets the mutation, and that's not very good for survival, right? So every now and then, in times of plenty, it's good if you outbreed, if you mix the genetics of different colonies. And that's what these ones are for. You get these big, fat, and brave because they go out of the colony, they brave the eagles, and they try to find either another big one, and the two of them mate, and they start a new colony. But then you don't want a colony of just big, brave, fat dispersal morphs, right? You want to restore the original colony order. A little bit like ants. You want one queen, you want a bunch of soldiers, and you want tons of workers, right? You don't want to suddenly have all soldiers and nobody working, right? So same thing happens in mammals. It's bizarre. Out there in nature, we get really everything. So in the lab, we see this exact same thing. Not big, fat, and brave, but we do get big and quite chubby. So these two are genetically identical brothers that are not at all artificial. They have a mutation in one copy of one gene. So they are much more normal than any two of us are, as far different, right? Between any two of us, there's thousands of mutational differences. Between these two, it's one base pair on one copy of DNA. That's the only thing that's different, and you can see the massive difference, right? And it's purely done to epigenetics. So these are their growth curves, right? So this is what normal mice would look like from 6 weeks of age to about 16. They get about 5 grams of fat, about 35 grams total, right? And so this is our colony. This is the one base pair that's been changed on this gene. And you can see we get 2 different types of offspring that come out. Normal ones and gigantic, and we call them gigantic. Gigantic because they have a much bigger lean mass and quite a bit more fat. And these two are identical twins, okay? Those 2 sets. It would be me being about 175 pounds, and if I had an identical twin brother that was 225. That's crazy, right? Unbelievable. And the idea that each of us could have come out A or B, right? So there's 2 types of variation, right? There's this switch, but also within each group, there's a bunch of variation. So that's all epigenetics. So what about humans? Well, you guys will be familiar with this data. These are boys, all Caucasian, born in the US, 6 to 8 years old, prepubescent. Most of them, as you can see, are skinny, and some of them are heavier. And you can see that the distribution of the data has 2 curves under it. This is already an inkling that could there be 2 forms of us? I mean, are there 2 types of families, the ones that eat heavy and the ones that don't? Are there 2 types of genetic blueprints? No, there's not, right? We don't have 2 separate populations. We have a mixture, right? We have a mixture of social backgrounds, a mixture of economic backgrounds, mixture of genetics. So we shouldn't have a curve that's got 2 bumps to it. We should have kind of a messy curve. That would be a mixture of many different conditions, et cetera. And we see this across, so this is all data from NHANES, which is the National Health and Nutrition Survey of the United States. 5 ethnicities are documented. We see this kind of bistable curve, we call this, across every single ethnicity. It's another argument that it's not genetic, that this applies to every socioeconomic group. And what's really interesting is if you look at historic data, so sort of from the 60s to the 90s, this little bump here was very small, and now it's much bigger. Something happened around the 2000s where, I mean, it could be smartphones and computers and that, but I don't think so. But what's interesting, in the popular press, you'll read that the whole population is getting fatter, right? That we're all getting more obese. But it's not true. If you read about the childhood obesity epidemic, because it's there. There are way more kids that are obese than used to be. But here's the data, is the skinny kids are exactly the same as they used to be. They haven't changed in 50 years. And the heavier kids are also at the exact same spot on the curve that they used to be. There's just more of them. So the whole population is not getting more obese. It's simply we're getting a bit less of one and a bit more of the other. And I think that's really, it's an important takeaway. And it raises a bit of a problem with modern medicine. Is in modern medicine, all our research, we do 8 replicates of the experiment, and then we take the average, and it's like the average is golden. Everything is about the average, but almost nobody is average, right? We're treating humans where everybody is different. We all get different genes, and we've all got different epigenetics, right? So can we control this stuff? Well, that's a good question. In humans, I guess the Dutch hunger winter shows us in theory it is changeable, it's modulatable. There's a beautiful mouse example. This is a natural mouse. It's called the agouti viable yellow mouse. So mouse research started from circus people trying to find the fanciest mice they could. So the original laboratory mouse is called the fancy mouse, right? And this is one of those fancy mice. These are all brothers from the exact same litter. They're all super inbred. They're all genetically identical. Look at their coat colors. This is like the cat, the calico cat, right? That's also epigenetic. The colors on calico cats, the cells that are brown and the cells that are white and the cells that are black, they're all genetically identical, but they flipped into different color states, right? Same thing has happened here. What's really interesting about the gene where this has happened is it's epigenetically regulated, but it also controls body weight. It also controls how much the animal eats. So if you look, the brown mouse that you can hardly see is quite skinny, and the mice get heavier and heavier as we go to the left. So not only do they become yellow, they become overweight. And what researchers have done is try different diets. So if you put high methyl donors, don't worry about this, what they are, during pregnancy, you get more brown mice. So what does that mean? You get more lean mice, which means you get more mice without diabetes, because these ones become diabetic, and you get more mice without cancer, because obesity is one of the biggest risk factors for cancer. Right? So the chain reaction of getting slightly more brown mice is you get way healthier population, right? From many, many different angles. Okay, with our research, I'll stop boring you there. Tara did ask me to talk a little bit about what's different about science today, because you're teaching the future generations of science. And I was thinking about this morning, it's very interesting. You know, the scientists I have that come into my lab are all 25 to 28, right? So I see them kind of 20 years after you do. So the ones I'm getting now are already so different than they were 10 years ago, but they're the product of teachers doing their work 20, 25 years ago. As long, you know, there's a big stack. So what's happening now, we're not going to see the scientist consequence for another, 15 to 20 years. You can imagine from the image I picked some of what's different now. The world of science is definitely changing, and it's harder to comprehend. And I'll give you a couple exhibits. So now when I did my PhD, I mixed things in reaction tubes and actually watched colors change and stuck things on a spectrophotometer that gave me one value that came out, and I wrote it and I graphed it. Now this is what data looks like. Now we put a sample on a machine, and it gives us 50 million data points, in 10 minutes, and that's what the current young, brand new scientist coming out of undergraduate has to deal with all of a sudden, right? So they look at this. They play with. They're on the computer all the time. This is already highly pre-processed data, so it's not even the raw. The raw is just digital sound bites. This is the kind of thing. This is nobody can guess what this is. I presume this is a snapshot of all the genes in the genome. But how do you look at 25 things and their changes at once? Well, we have to come up with some way of visualizing it. So we put it in 2 dimensions, and we cluster. The purple genes are all the same type of gene, right? The same type because they're in the same type of chromatin, right? In the same epigenetic state. And then on top of that, we can layer how those genes are changing when we do something to the system, right? That's what this would be. So the red color means all the genes are turning on, right? These genes are being upregulated. So this is the kind of data interpretation that students have to be ready for, and they have to be able to do this on their computer terminal, coding it, in R at the moment. But hopefully that'll all be simpler in the future. So I think one of the biggest things, in addition to having to require different educational modalities that they are really comfortable embedding themselves in and always taking in new information, is this aspect. And I use the cliche, there's no I in team. Modern science is a team sport. There's no way you can do it on your own. It's impossible. Just to tell you what the data was here. So this is what happens to your whole genome when you get obese, right? The genes that are active, those are down here, don't really change at all. What happens is the silent genes, the genes that aren't supposed to be on, start turning on. Interesting. We don't know exactly what it means or what the consequence is. So yes, Modern science is team science, and that won't change. And that happens in the lab scenario, that if you have a lab of 2 or 3 people, it is very difficult to do research. It happens at an institute level as well, though. If you don't have the right colleagues that have the right teams, the right networks, and the right access to resources, whether it's intellectual, whether it's the networks, or whether it's $1-million microscopes, it's very hard to move forward. And that's why this place is successful. It's full of fantastic people. Like Tara said, if you've got the opportunity to come yourself or bring your students here, please do. So this is just a portion of our faculty. This is the Center for Epigenetics. Gosh, the autocorrect, there's an S on it, just that you know. There is an S on it. Yeah, and we've got real pioneers of the field here. Peter Laird does cancer epigenetics. He was one of the leaders of TCGA, the Cancer Genome Atlas. This is researchers all across the country mapping hundreds or thousands of cancers that we can figure out what makes a cancer definitively harmful, what makes them not harmful. There's no way that even an institute could do that alone, right? This needs tens, if not hundreds, of institutes. So, on that note, science is a very different thing. Is this a turning point? I don't think so. I think it's repeating history. I think, if this talk was being given 20 years ago, the last person standing up here would be saying, oh, wow, the new students coming in, they've got such a challenge. They have to deal with all of this new information. Part of that is science. You're always on the frontier. You're always dealing with the unknown. So you're always learning the unknown, and you got to love it. You got to bask in the anxiety of not knowing what's going on and trying to figure out the puzzle. Like that's part of the process. So if you've got people that love learning, that come to school, love learning, and love that next new idea, those are the future scientists. They'll love it. But nowadays they have to be able to do this. They have to talk. They have to be able to communicate, and particularly because of the team aspect, it is now a real struggle to do anything on your own. Okay. Yes, so I guess that's the conclusion. Hopefully I've given you some food for thought on who am I and who could I have been, and hopefully it gives you a new perspective on each of your students. And yeah, thanks very much for your attention.